Alright guys, thank you for tuning in one more time. The subject of this lecture is what would have been our fourth lab, where we did some DNA isolation sequencing. So it's essentially what we did today in lab. I'm going to talk about the theory behind DNA purification and how we actually sequence the DNA. How do you take a piece of DNA and figure out what the A, T's, C's, and G's are in that sequence. So, the plates that you made today, this evening, I'm going to come in the morning, and they're mostly all going to look like this. You're going to see a round agar plate. There's going to be a few colonies on there. So how do we find out if those colonies actually have your gene? Remember, we're putting your gene into a plasmid that had another gene before you digested it. So there's always a little chance that the colonies you see here on this plate could contain either your new gene or the old gene. Hopefully we've done everything right and it's the new gene, but we still need to check that. And anytime you work with DNA, there's always a slight chance that there's going to be some kind of mutation in the DNA. It's always a possibility. Remember, we look at those agarose gels under UV light, and UV light is a mutagen. So, at any rate, here's some things that we can do to verify that these colonies have the DNA that we want. First of all, we can do a colony PCR. We'll talk about that later. That's where we actually use the bacteria themselves as template DNA for a PCR reaction. It's a quick way to determine if the gene is in the cells or not, as opposed to the old gene. The next thing we need to do is take one of those colonies and prepare a culture. So we have here a 50 ml tube with about 5 to 10 mLs of liquid broth. So this is pretty similar to what I gave you today. Remember, you had about 5 ml of that liquid culture, and that's what we isolated the DNA from. So here we go. You take a little colony there, use it to inoculate some broth, and you get a starter culture. And once you have that starter culture, it's important to make a glycerol stock. So all you do there is take some of the bacteria, mix it with 50% glycerol, and that gives you a mixture that is safe to freeze. The glycerol helps the cells tolerate the freeze-thaw process, so you want to save some of those cells for posterity. If you ever want to make more of this DNA in the future, you want to make sure you have a glycerol stock of those cells. And then we do what we did today. We take the remaining cells in that starter culture and purify the plasmid DNA from them. And once you have that purified plasmid DNA, we go on to sequencing. So, here are a few things that, well, I guess we kind of generalized in the lab today, and otherwise you would have done in the 2 labs that we missed for snow days. And again, the sequencing is very important because it checks to make sure that all of your bases are where they should be. So not just is it the new gene or the old gene, but is the new gene completely accurate in its sequence. Okay, so let's talk methods. If you've got your colonies there on the agar plate shown on the left, all we need to do to start a starter culture is, first of all, make media. So anytime you're growing E. coli, LB media is a pretty safe bet for growing these cells. LB stands for Luria bertani. There's 2 people that invented the mixture. It's just a mixture of some pretty simple stuff. You have their yeast extract. That's what the bacteria use as a carbon source and for vitamins. Tryptone. Now, a tryptone, in case you are unfamiliar with the term, that's just a bunch of proteins that have been broken down into amino acids. So a lot of your vitamins are just essentially amino acids. So tryptones supplement the yeast extract. And then we have sodium chloride. Now, different bacteria require different amounts of sodium chloride. E. coli like it pretty high, actually. So you can see there we've got 10 grams per liter of NaCl. So anyways, you can mix all of this together on your own. You can buy yeast extract, tryptone, and NaCl, or there's plenty of companies that offer pre-mixed powders for this. Either way you do it, you add water, autoclave it, and you get media, the brown solution shown in the tube on the right. And then all you have to do is light that Bunsen burner, make the bench air nice and sterile, go down, touch a tip to the colony, and transfer that tip to the liquid. All you need to do. But it's very important here that you make sure that your tips are sterile. Otherwise you'll contaminate that starter culture. So autoclave your tips and your media at the same time if you can. Oh, one more thing. We have to add ampicillin to our media. That makes sure that the cells that we take and put into the media will maintain the plasmid. It doesn't ensure that it's the right plasmid, but it does ensure that those cells that are growing in that media have our plasmid DNA. And then all we have to do is take that tube, put it at 37 degrees C in the shaker for about 8 to 16 hours until it becomes nice and turbid. And that's the point at which I gave you the cells today. So you had that tube, nice and cloudy, lots of cells. That's what you want. You want a lot of cells because that means you're going to get a lot of DNA. And like I said before, the very first thing you do when you have that culture, we didn't have time to do it today, but I did it before you guys came in, is to make a glycerol stock of those cells. So you're going to take 500 microliters of your culture and mix that with 500 microliters of 50% glycerol. And again, the glycerol is just to help the cells tolerate the freeze-thaw process. They can also use it as a carbon source if they get desperate. And then you throw that directly into the -80 freezer. So keep these cells in cold storage. Not the -20, keep them in the -80. Once you have your glycerol stock done, you just centrifuge the cells. So from here on out, it's just a review of what we did today. 3000 G for 5 minutes usually works. We did 4500 G for 10 minutes. Either one. Alright, then you get rid of the old media. First step in purification, get rid of your old media. Resuspend the cells in a resuspension buffer. I have it here called R3. I think that's what Kaijin calls it. And here you just want to resuspend in enough buffer to get all the cells up. So like today we had to add more to get even more cells up. That's going to be a judgment call on your end. Typically, as a good rule of thumb, I like to double whatever the company recommends. So you get a protocol from the company that you buy a kit from. If they recommend 200, go up to 400. You'll get higher yields, trust me. I think lysis is just more efficient when you double everything. So at any rate, you have your cells resuspended in that tube, and then you add your lysis buffer. And now remember, the way we lyse these cells is called alkaline lysis because we raise the pH up to around 10 to 12. And that's enough to completely destroy any cell membranes and cell walls and protein. But the DNA is able to survive that. So we're wrecking everything else except for the plasmid DNA. And then we add neutralization buffer because if you let that go on for long enough, it will eventually wreck the DNA. And that's when we get that nice white cloudy stuff, that white snot that we saw today. And then all you have to do is centrifuge that mixture to separate the snot from the liquid that contains the DNA. One more note here. You want to always treat the snot and all of these mixtures very carefully. Don't vortex them. If you do, it'll shear the bacterial chromosomes, the genomic DNA, and you'll find that then in your final product. The way these kits work is they separate the DNA, the plasmid DNA, from the chromosomal DNA based on size. So up here when you do lysis and later on when you bind the DNA to the resin, that's all based on size. Bigger things don't work as well, smaller things work better. So don't vortex it because then you'll disrupt the genomic DNA into smaller pieces that will co-purify with your plasmid. Alright, so once we have that cell lysate there, all we have to do is transfer that to the column. Now, some kits, like the one we used today, will have you use a column preparation solution first. So you add that, spin, and then transfer in the lysate. But other kits do not require this step. So just follow your protocol. And once you do that, now we'll have DNA on the resin, which is the gray bar shown there in the tube. and the waste that flows through. So this is all the liquids and other impurities that might have remained after lysis. We wash once and wash twice. That's just to remove anything else that might be bound to the resin, and now we should have nothing but DNA bound to those columns. And at that point, we can transfer that column to a new tube. We don't want to use the old tubes. They're contaminated with ethanol and whatever proteins might have come through there. So we transfer the column to a new tube, add 30 to 50 microliters of water, and spin one more time. And that gives us our purified plasmid DNA. Now, if you want a higher concentration, use less water. If you want more yield, use more water. That's about how that works. And then we quantify. So we did this as well. So we did this today. We quantified the DNA using my plate reader. And you might remember I mentioned a machine called a nanodrop. And here's what the readout for a nanodrop looks like. It's the same thing. You have a little pedestal, just like we did today, a little plate of glass that you load your 2 microliters of DNA onto, and then it reads the absorbance of it. And the nanodrop's pretty nice. It's got a nice user interface. It gives you the spectrum right here. So you can see there that peak at 260, that's your DNA. You want a high peak at 260 nanometers. DNA absorbs at that wavelength. And then lower everywhere else. And just like the plate reader I used today, the nanodrop calculates your concentration of DNA. And that's what's shown in the green box down there. So here we have 182.5 nanograms per microliter. And it gets that concentration just using the Beer-Lambert law, A equals ECL. So we know what E is for the DNA, so it's easy enough to calculate concentration based on absorbance. And just like my plate reader today, any good DNA quantifier will kick back 1 or 2 different readings as well. You have your 260 over 280. This lets you know if there's any protein, single-stranded DNA, or RNA contamination in your sample. So if the absorbance at 280 is high, that means you're going to have a lot of protein that you don't want, and then that ratio there is actually going to go pretty low. We ideally want it to be around 1.8, a little bit less than that. Then we also have another ratio, 260 over 230. Now there's carbohydrates, EDTA, and phenol. Those all absorb around 230. So sugars from the cell, some solvent contaminants could get in there. There again, we want the number to be around 1.8 to 2.0. If these numbers vary too much from those numbers, then you probably have some contamination. Something went wrong with your purification, and it may or may not be a cause for concern. If you try to use this DNA that you purified for a reaction and it doesn't work, and your numbers, your 260 over 280 or 260 over 230 is off, that may be why. You might want to repeat the purification to clean up the sample a bit. Okay, so let's say you do your DNA purification, you get a nice high concentration. The next step then is to send your DNA out for sequencing. Some universities have in-house sequencing machines. I'm not sure that if Villanova has one. I don't think we do, but that's no problem. There's plenty of companies, actually even plenty of other universities in the area. For instance, University of Pennsylvania has a sequencing facility. So you can send it to UPenn or any of these companies, and they will sequence it for you. Fairly common procedure. Alright, so all sequencing is just a modified PCR reaction. So there you go. What is that, about the 4th or 5th time that I've mentioned a modified PCR reaction for some kind of purpose? PCR is a fantastic technology. We use it for so many different things. In this case, what we're going to do is, just like normal PCR, we're going to take your template DNA, melt it, so we get nice single-stranded templates. And we're going to add a primer. Now in this case, we're not really trying to amplify genes or double-stranded DNA. We're only looking for a single sequence. So all we do is add one single primer. You don't need to add 2 primers, just one primer. Just like normal PCR though, that primer binds, and we get extension. So we have a Taq polymerase that starts copying the template. However, the twist here is that in addition to all my normal base pairs, so we have the C, T, G, and A up there in the top right, in addition to these normal DNTPs, we also have DDNTPs. Now what a DDNTP is a dideoxy nucleotide triphosphate. And that dideoxy means that these guys will join a growing DNA strand, but they can't continue that growth. And they can't continue that growth because where the 3' OH would be, we instead have a fluorescent tag. So the Taq polymerase still grabs onto these modified bases and integrates them into the growing strand. But then it terminates. So here we have a new DNA strand that's 9 base pairs long and ends with a yellow fluorescent tag. So the DNA synthesis ends right there. We can go into another cycle for PCR, and a similar thing happens. Except now, instead of the G ending it, now we end at C. So what's happening here is these fluorescently tagged DDNTPs are randomly inserting themselves into these PCR products, such that you get a combination of different lengths of PCR amplified genes. Some might be 5 base pairs long, others might be 10, 20, 21, 22, 23. You repeat this enough times to get many, many different copies of each different length. And you'll notice here, so all the C's are going to be red, all the G's are going to be yellow. So we have a nice mixture of different lengths of PCR products, and each corresponding length has a corresponding color. And so we can say if this 20 base pair long template is red, it must end in a C. If it's yellow, it ends in a G. So keep in mind, at the end of this reaction, we have a mixture of all these different PCR products, but they're still in the same tube. So how do we separate them? Well, we need a sequencer for that. Now what a sequencer is pretty much just a complicated or a complex agarose gel electrophoresis system. If you look over here on the right, you'll see these tubes, very small tubes. They're actually called capillaries because this is a capillary electrophoresis setup. And the benefit to capillary electrophoresis is that I can fill these little tubes with gel, agarose gel, the same gel that we use in our gels. And that gives me an extremely long gel, very narrow, but extremely long. And it's long enough to separate all of my PCR products from the sequencing reaction by a single base pair. So my sample enters those capillaries and spreads out. And then it enters this detector, the black box that says danger. And so, as you might expect, the smallest piece of DNA comes through first. So that might be my first few base pairs of the sequence. So let's say my smallest product is 20 base pairs long. It enters that detector, and if it's a yellow piece of DNA, then the sequencer knows, oh, that's a C. If it's a red for the next one, it says, oh, base pair 21 is a G. And then blue is a T, and yellow is a C. And it puts all this information together to give you this readout shown here. So you can see in this example here, my first product was a T, so it shined red, and we have a little peak there. And then the next one was blue, and so the sequencer knows it's a C. And so it goes through hundreds of these base pairs, and it's detecting each one of these PCR products of a different or a progressively longer length has a different color, means a different base. So the sequencer runs through all these. The whole process is actually pretty quick. It only takes a few hours. Some sequencers now can do it even faster than that. But but it gives you this nice readout, and this is perfect. This is just the kind of readout you want to see. The company is going to show this to you as part of the data that they give you, and you want to see these nice defined peaks that you see here from bases about 1 to 380. What you'll notice as we get farther on in the sequence that my resolution starts decaying a little bit, especially down here around base pair 760. You see those peaks aren't very well defined, and I start getting a lot of these light blue ends in my sequence. Now what an end means is that the sequencer can't really discern what kind of base that is. It might get a little bit of red, a little bit of blue signal, as you can see in some of these cases, but it can't really tell, is that a T or is that a C? So most sequencers have a window of about 1 to 800 bases that they can confidently measure or determine the sequence for. Once you get past that 800, then you start seeing a lot of ends, and you can't really trust that data. So if you have a really long gene that you want to sequence, you're probably going to have to do multiple overlapping sequencing reactions. And this technology might get better in a few years. Maybe you can sequence up to about 2,000 base pairs, but this is about where it is right now. Now, once you have all that data, all that sequencing data, the company will probably send you a notepad file or something like that that's got your T's, your A's, your C's, and your G's. You want to check that or compare that to your predicted sequence. So if you look in your lab manual, you can see the expected sequence for each of your genes. That's in your appendix. So you would take the data that they send you and compare it to what I have in the appendix to make sure there's no mutations in the genes. And what I have shown here is a sequence alignment tool called BLAST. So you can click on that link right there and go to this website sponsored by NIH, National Institutes of Health. And what it allows you to do is it has two fields. And you copy and paste your sequencing data in one field and your plasmid sequence or your gene sequence in the next field. And then it gives you this nice alignment right here. So it aligns one sequence to the next, and you can read through this and you can see every vertical bar is a positive match. So here we've got about 100% sequence alignment. This is what you want to see. Sometimes you might see an N in here. If you see an N, don't be scared. That doesn't conclusively mean that there's a mutation, but there might be. You might want to repeat the sequencing reaction to resolve that. But if you see, let's see, a C where a G should be, then it's you're pretty confident in saying that there's a mutation there. And so something happened to your gene, and that may or may not affect the protein that you express and purify from this plasmid. If you see a mutation, it's probably a good idea to start all over. Or, if your transformation yielded several different colonies, maybe you go back and you try a different colony. So you don't have to repeat your PCR digest and everything else, but you just use a different colony for sequencing. For that reason, whenever I make new plasmids, I'll usually pick 2 to 3 different colonies, purify the plasmids for those, and send those all off for sequencing, and then pick the one that looks the best. Hopefully you won't see any mutations, but it's nice to have backups like that. Okay, so sequencing is very nice. It's a very good technique. It tells you exactly what's in the plasmid. But let's say you're in a little bit of a hurry. Maybe you don't really care if there's mutations in your gene. Maybe you just want to make sure that your gene is in there and proceed with your protein expression. If you want to do that, there are some shortcuts available. This one right here is called colony PCR. So let's say you come in the morning, you have a plate, and you got about 10 colonies on there. Using colony PCR, I can tell you conclusively which one of those colonies has your gene within about 2 hours. So all you do here is you make a master mix for PCR. So just like we did in the first lab. You add your primers, Taq polymerase, your buffer, nucleotides, and magnesium. All that good stuff, you mix it together. But you do not add any template DNA. Instead, what you do is you make that master mix, and then you split it into a bunch of different tubes. So let's say you come in, you have the plate over there on the left, and it's got 10 colonies. You're going to split that master mix into 10 tubes plus another one. So 11 tubes, and that 11th tube is for your positive control. So in that last tube there on the right, we're going to add our template DNA. So this is our positive control. That just means that solution contains the gene that we're trying to amplify. So we just have to have that positive control in there to make sure that everything works with the PCR. And then to add template DNA to the rest, the other 10 tubes, what we do is we take a tip, touch the colony, dip it in one of those tubes, and then put it in some LB broth. So what we've done there is we've used the tip to add just a few cells to one of these tubes. And those few cells are enough template DNA to fuel a PCR reaction. So if we have 10 colonies, we take up one colony, put it in the first tube. Take another colony, put it in the second, third, and the third, so on and so forth. And you want to save those tips because one of those, or one or more of those, will have your gene that you're trying to clone. So you set that up, you have template DNA and everything in there, and then you run it just like a normal PCR reaction. And these are some of the results that you might see. So here we have these 4 quadrants, upper left, upper right, so on and so forth. These are all different colonies from a plate. Now what we're looking for here is a positive signal. So we want a nice bright band there around the middle of the gel. So you can see in the bottom right we have our positive control. This is our plasma DNA that we used for PCR and digest. Here we have a nice bright band there in the middle of that lane. So, as I have shown up here in the upper left, that yellow circle, there's no band in those colonies. So those colonies have a plasmid, but it doesn't have your gene in it. It might be the original gene that we removed with the digest. It might be something else altogether. We don't know. But it doesn't have your gene. Now the lower yellow circle, we do see a band. So those colonies you can trust. They do have your gene in them. So you would take those tips that you saved for those colonies, grow those up, and sequence those plasmids. So at the very least, you know that your gene's in those colonies. You don't know if there's any mutations or not, but it's a good place to start. So there you go. That's about all I have for this lecture. I promised you it'd be pretty quick. So remember, our test is going to be on Monday. No notes, or closed notes, closed book. Study everything. Study the PowerPoints. Make sure you know how to troubleshoot your reactions. If something goes wrong, what can you do to fix it? Likewise, know how to set up reactions. Yeah, and that's about it. Feel free to email me if you have any questions. And remember, our lab report is due not next Wednesday, but next Friday. So that last day before spring break. Please get those to me by 5 p.m. on that Friday. Alright guys, thank you very much, and I will see you on Monday.